students, today we are going to look at the topic on enzymes. So enzymes, this topic is pretty much quite straightforward. Usually the hard part comes in when it comes to the writing. Okay, so it follows a very strict answering technique which we will look at later, and the questions are pretty much straightforward. So let's start off with this part here. You do have to know the definition by heart that they are biological catalysts, not just simple catalysts, but it must have the word biological inside. And they are made up of proteins, so bear in mind for this part that it is made up of proteins. So in other words, organs that synthesize high quantities of enzymes are required to have high amounts of rough endoplasmic reticulum and Golgi body to be able to produce the proteins, which are the enzymes. If you are not sure what is the RAR and Golgi body, then refer back to the topic on cells and structure. So second part to this one, it is highly specific and it is used to speed up the rate of the reaction without being altered at the end of the reaction. So this lack of alteration is of course dependent on the optimum pH and the optimum temperature as well. Okay, so when we take a closer look of it, properties of enzymes, make sure you know at least 3 to 4 of them by heart, because sometimes they can come out in open-ended questions to list the properties. So we have seen that it does have a 3-dimensional configuration and is made out of proteins. So we also saw that it speeds up the rate of reaction. So how does it speed up the rate of reaction? It lowers the activation energy. So once the activation energy is lowered, that means to say that amount of energy needed for the reactants to actually start the reaction. Okay, originally this is the amount of energy that is needed, but in the presence of the enzymes, this energy is lowered. So once the energy is lowered, that means to say that the reactants are not required to take in as much energy. So subsequently, the reaction can be much faster to form the products. Okay, so bear in mind that in no way do the enzymes change the quantity of the products formed. So it is still going to be the same amount of products with or without enzymes. It is just the speed, the time taken to form it. Okay, so enzymes, they are also required only in minute amounts, not minute amounts, because they are not changed, so they can be reused. Of course, this is subjected to optimum pH and optimum temperature. So due to its specificity, we are using this thing called the lock and key hypothesis. So IP students, you'll be looking at the induced fit hypothesis for some of the IP schools. Okay, if your school, even if it is IP, does not do the induced fit hypothesis, then by all means you can just keep it at the back of your head. Don't have to go into detail. So this lock and key hypothesis, often make sure you know how to draw it or at least sketch it out. It doesn't have to be as intricate as this. So you can just draw out some rough shape, but make sure that the grooves, which represents the active sites, along with the substrates, they match each other. Okay, so you can just draw something simple, maybe like one is a square, the other one is a circle. It doesn't matter as long as the grooves, they match. So subsequently, you have to label that this is the lock, which is the enzyme, and the substrates, which are the key. Okay, and then they form this thing called the enzyme substrate complex. So label your drawings enzyme substrate complex, identify which is your enzyme, which is your substrate, and name your enzyme and your substrate. Subsequently, when they form the products, make sure that your products, they are joined together if it is an anabolic reaction. So anabolic reaction means from small things you are building up. So for example, from the amino acids in your body system or the glucose in your body system, it then subsequently build up to become polypeptides or maltose or even glycogen. Okay. Catabolic, on the other hand, it is breaking down. So same thing, you have got your enzyme, you got your substrate, and then it binds together, and then two substances get produced. Now this one is a bit tricky, because if you are doing maltose, for example, if you go to the nutrition topic, if you are doing maltose, then you get 2 glucose, and make sure your 2 glucose must be identical, because they are after all glucose. But if you are doing, for example, a polypeptide, then you have a whole string of amino acids, and these are going to look different. Okay, so be very careful of the products that is being released, the shape of it, because it can be penalized. Now, as we move on to here, so lock and key hypothesis, the specificity is due to presence of the active site. Okay, so only specific substrates have this complementary shape to the active site, and once they form this ESC, which is called the enzyme substrate complex. So once it fits into here, doesn't mean that it just stays stable. There's a lot of chemical reactions that take place within this three-dimensional conformation, and you have a conformational change which will then release the substrate, which will then convert the substrates into the respective products. Okay, so once the products are released, the active site will go through another change, okay, and then subsequently it will then carry out further reactions. Slight change, not a major change, because in order to bind these two together, there is a slight conformational change, or to separate this one. Okay, to bind these two, there must be a slight change to ensure that the bonds are formed between the new substances. So when we take a look at the effect of temperature and also the effect of heat on enzymes, this is similar to each other, synonymous. The other one is the effect of pH on enzymes. Okay, you will see that the answering technique is very vital. So your effect of temperature or effect of heat, how do you differentiate the graph compared to pH? For temperature, low temperatures, it will never go to zero. Okay, enzyme activity doesn't mean that it is totally denatured at low temperatures, but at very high temperatures, it can be completely denatured. So the spectrum is very broad. The base is very broad. pH is less forgiving. Okay, you can see that the base is much lesser. For IP students, some schools do the terms protonation and deprotonation. If your school doesn't, then same thing, by all means, just put at the back of your mind. Okay, so the effect of pH on enzymes is more acute due to the deprotonation and protonation impact. So coming back to the temperature, so this part over here is what we call below optimum temperature. And this one is the optimum temperature, and this is above optimum temperature. So why do I keep repeating the words optimum? Because you have to use the optimum temperature as the benchmark. So refer to 3 pointers. First part here, second part here, and the third part here. So first part here, below optimum temperature, low kinetic energy, low frequency of effective collision, so low enzyme yield. Subsequently is optimum temperature, and then as the temperature increases beyond the optimum temperature, there is the loss of hydrogen bonds. So all these three-dimensional shape is held by the hydrogen bonds, which then keep them in place over here. So once the hydrogen bonds are lost, it cannot be held together, so it becomes a linear structure. So as the shape increases, as the temperature increases gradually, more and more hydrogen bonds will be lost. So once the conformation at the active site is lost, that's when the substrate can no longer bind. Okay, so this change is irreversible and is said to be denatured. The enzyme is said to be denatured. Now pH. Okay, so similarly, you have got below optimum pH, and above the optimum pH. Okay, so denaturation is irreversible for pH on either spectrum because of changes in the hydrogen bonds and changes in the amino acid structure. Okay, now even though this one says IP, it does come out in application questions, so it's pretty straightforward. Okay, over here, enzyme concentration increases. Ideally, the rate of reaction should be to infinity, but this does not happen because as the substrate concentration increases, there is a finite number of enzymes. So eventually it reaches what we call the point of saturation over here. So increment in substrate will no longer increase rate of reaction. So the only way is to increase enzyme concentration, which in the human body system, we are more like a closed vessel, so it's not too feasible. This application by increasing enzyme concentration to increase the rate of reaction is more applicable for industrial purposes. Okay, so induced fit model, more for the IP school. So enzyme substrate complex, as the term suggests over here, is more of your enzyme and the substrate in the lock and key hypothesis, whereby it is one lock to one key. So after some time, they realize that there is no such thing, because if I have 5,000 substrates in my body system, then I'm expected to have the same 5,000 number of enzymes to fit the 1:1 ratio, but it is not the case. So the human body system or mammals body systems, the number of enzymes to the number of substrate is not 1:1. So this is whereby the induced fit model was then developed, whereby substrates that are very, very close in conformation to the active site. Okay, as the substrate goes closer and closer to the active site, you can see that these two doesn't match totally, but it is very similar. Okay, this active site will then undergo a conformational change, slight minor change, to be able to fit the substrate. So of course, if my substrate is a circle, okay, there is no way that my active site can undergo a huge major change, but minor changes is still fine, which is why it is called an induced fit model. Okay, so competitive and non-competitive inhibitor, I'm just going to go through this briefly because it's not a very commonly asked question. It can come out as an application question for some schools. So my competitive inhibitor, as the name suggests, it competes for the same active site. You can see that the shapes are very similar. So for example, the competitive inhibitor carbon monoxide competes with oxygen molecule to bind to hemoglobin irreversibly. So it competes for the same active site. So once it competes ready and binded, is unable to bind. Okay, non-competitive inhibitor on the other hand, it is totally different. So what it does is this non-competitive inhibitor will bind to a site that is away from the active site. So once it binds away from the active site, this active site will then change the structure. So which means to say that the original substrate is very hard to bind already. Okay, so that's it for enzymes. Thank you for watching.